So in the past video, we talked about the photosynthesis. So we learned that photosynthesis is the food-making process of plants. This process occurs for the plants to produce their own food. They make glucose, and this sugar is considered as the product of their photosynthesis. Other than that, they also produce byproducts such as oxygen. There is a question: do humans also produce their own food? Where do they get their energy for doing such activities? Humans exist to do a certain job. Like plants, they need food to finish their job. But where do humans get their energy? So, humans are considered as heterotrophs. What is heterotroph? Heterotroph, an organism that cannot manufacture its own food and instead obtains its food and energy by taking in organic substances, usually plant or animal matter. So, because we are living things that cannot make their own food, instead we get our food by consuming other organisms, which why we were called consumers. So it's clear now that humans cannot produce their own food. Humans consume other organisms as a food. So the big question here, how these food are converted into energy. The conversion of food into energy has a process, and it is called as cellular respiration. To fully understand what is cellular respiration, let's first define what is cellular. In biology, cellular is relating to or consisting of living cells. Living cells means eukaryotic cells. So eukaryotic cells can be found in both animals and humans. So the next is respiration. What is respiration? Is the process by which living things convert oxygen and glucose to carbon dioxide and water, thereby yielding energy. It does not require the presence of sunlight and is always occurring in living organisms. It takes place in the mitochondria of cells. It has two main types, aerobic respiration and anaerobic respiration. So if we combine the thought of the two words, we can come up with the definition of the cellular respiration. What is cellular respiration? Cellular respiration is the set of reaction that produces ATP. ATP, quite possibly the most talked about molecule in biology, is our energy currency. Cellular respiration uses glucose to produce ATP our body needs to perform essential functions. Cellular respiration can be divided into three parts: glycolysis, the citric acid cycle or the Krebs cycle, and the electron transport chain. So, as what I have said earlier, that the cellular respiration is divided into two types. Let's first discuss the first type, which is the aerobic respiration. So, from the word itself, aerobic means oxygen, or aero means air. So, this process or the phases under aerobic respiration needed oxygen to continue the whole process. So, what is aerobic respiration? These phases are nothing more than metabolic reactions involving the conversion of glucose and other molecules into carbon dioxide and water. The resulting energy released from this conversion will be used to produce ATP. So here in the picture, you can see the presence of oxygen in both land and water, which defines that oxygen is essential for living things. So we have here the four phases of aerobic cellular respiration: the glycolysis, transition or acetyl-CoA reaction, Krebs cycle or the citric acid cycle, the electron transport system or electron transport chain. So these four phases need oxygen to continue the process. the first step of cellular respiration is the glycolysis. So what is glycolysis? Glycolysis occurs in the cytoplasm, specifically in the cytosol. So reactants, we have here the glucose, 2NAD+, 2 ATP, 4 ADP, and 4 inorganic phosphate. And we have here the products of 2 pyruvates, 2NADH, 4 ATP, a net of 2 ATP, and 2H2O. And here we have the chemical formula for the production of or the conversion of glucose to ATP energy. So you can see here at the left part of the are the reactants, which are the C6H12O6, and at the right part are the products. So we have here at the end the ATP energy. So the glycolysis process was divided into two steps. The first, the energy investment step, and the second is the payoff step. So here we have the picture of the glycolysis process. So we have the first step in the glycolysis, which is the energy investment step. So we all know that the chemical formula for glucose is C6H12O6. This C6 or the 6 carbon will be break down into two groups. So this C6, you can see here the C6 will be break down into two groups. So why this first step is considered or called as the energy investment step? As you can see, there is a presence of 2 ATPs. These 2 ATPs was used to break down the 6 carbon compound into two groups. So after the splitting of glucose, the next step follows, which is the payoff phase or the early harvesting step. So the C6 was split into two groups of 3 carbon compound. You can see here the pigal, other call it pigal, other call it they have many terms for that process. For this picture, we have here the pigal or the phosphoglyceraldehyde. So it's really hard to spell. So let's back. So other called it pigal or phosphoglyceraldehyde. Each group has a phosphate attached to its ends. You can see here the capital letter P. Yeah, right. That is the inorganic phosphate that came from our ATPs, right? ATPs. So these phosphates came from the ATPs that was used to break down the 6 carbon. So that 2 ATPs that we use in the energy investment phase. So from the pigal, what are the products that we can get? So from this pigal process, we have here we produce two things, which are the NADH, 2 NADH, and 2 ADP. So from NAD plus, how this NAD plus became NADH. So we all know that in chemistry, reducing is gaining. So here we reduce the positive electron and become NADH. So from NAD plus, the NAD plus become NADH. We also gain 2 ADP. So this 2 ADP will gain another phosphate at its end. So from these two phosphate, so we have here the ADP from that will gain 2 phosphate at each end, and this ADP will become ATPs. So all in all, in this payoff phase, we get 2 NADH and 2 ATP. So this pigal will be reconfigured into pyruvate or pyruvic acid. Pyruvate or pyruvic acid. From this configuration, we also gain 2 ATP, again from ADP with an inorganic phosphate attached to it, it will become an ATP. So here, a water was produced from the configuration. So we have here 2H2O. And at the end of the process, or at the end of the glycolysis process, we can get the product of 2-pyruvate, 2-NADH, 4 ATP with a net of 2 ATPs, and 2H2O. So, as you can see in the previous slide that we must suppose, or we have a goal that we must get these products of 2 pyruvates, 2 NADH, 4 ATP with a net of 2 ATPs, and 2H2O. So at the end of glycolysis process, we attain the products from the glycolysis process. So that's all. We have another step, which is the transition or the acetyl-CoA reaction.
The next phase is the transition or the acetyl-CoA reaction. This reaction doesn't belong to either glycolysis or Krebs cycle. This is only an intermediate step. On the last phase, which is the glycolysis, it will end with the production of two 3-carbon pyruvate molecules, so these cannot enter the Krebs cycle, and what they do is decarboxylated to 2-carbon acetyl-CoA molecules. The reactants are 2-pyruvate and 2-coenzyme A. And the product will be 2-acetyl-coenzyme A and 2-carbon dioxide. There are also 2 molecules of nicotinamide adenine dinucleotide and hydrogen produced. Each molecule of acetyl-CoA can now enter the next cycle. The next phase is the Krebs cycle. The 2-carbon acetyl-CoA entering from the intermediate step leaves the cycle as 2 carbon dioxide molecules. This 2-carbon combines from oxaloacetate, which has 4 carbon molecule, to form the 6-carbon citrate molecule or citric acid. The next process is the decarboxylation, and it releases 1 carbon dioxide and will produce 1 NADH. So we have now 5 carbon molecules or the ketoglutarate. Another decarboxylation releases 1 carbon dioxide and another NADH will produce. The triphosphate is produced, then the substrate is oxidized, producing FADH2 or the fumarate adenine dinucleotide. The substrate is hydrated, then the substrate is oxidized again and producing NADH. And regenerating the oxaloacetate molecule, then the process will repeat again and again.
already done with the three other phases of aerobic cellular respiration, which are the glycolysis, transition acetyl-CoA reaction, Krebs cycle, and the last phase, the electron transport chain. So, let's first define what is electron transport chain. What is electron transport chain? The electron transport chain, aka ETC, is a process in which the NADH and FADH2 produced during glycolysis, beta oxidation, and other catabolic processes are oxidized, thus releasing energy in the form of ATP. The mechanism by which ATP is formed in the ETC is called chemiosmotic phosphorylation. So let's have further introduction with the electron transport chain. The byproduct of most catabolic processes are NADH and FADH2, which are the reduced form. Metabolic processes uses NADH and FADH to transport electrons in the form of hydride ions. These electrons are passed from NADH or FADH2 to membrane-bound electron carriers, which are then passed to the other electron carriers until they are finally given to oxygen, resulting in the production of water. As electrons are passed from one electron carrier to another, hydrogen ions are transported in the intermembrane space at three specific points in the chain. The transportation of hydrogen ion creates a greater concentration of hydrogen ions in the intermembrane space than in the matrix, which can then be used to drive ATP synthase and produce ATP, a high-energy molecule. So let's discuss now the whole process of the electron transport chain. In the diagram located below, there are the major electron transporters responsible for making energy in the ETC. So we have here the diagram of the whole electron transport chain process. So the first phase occurs in the intermembrane space. An integral protein that receives electrons in the form of hydride ions from NADH and passes them to ubiquinone. The second phase, the succinate dehydrogenase from the TCA cycle, a peripheral protein that receives electrons from succinate. Succinate is an intermediate metabolite of the TCA cycle to yield fumarate and FADH2. From succinate, the electrons are received by FAD, a prosthetic group of the protein, which then become FADH2. The electrons are then passed off to ubiquinone. So the next step is the ubiquinone to ubiquinol. You can see here in the diagram the letter or the capital letter Q. So from this ubiquinone, the oxidized form of the molecule receives electrons from several different carriers, from phase 1 to glycerol 3-phosphate dehydrogenase and ETF. It is now the reduced form which passes its electron off to phase 3. So the phase 3 occurs in the matrix, an integral protein that receives electrons from ubiquinol, which are then passed to cytochrome C. So next is the phase 4, which is also occur in the mitochondria matrix. So an integral protein that receives electrons from cytochrome C and transfer them to oxygen to produce water within the mitochondria matrix. And the last step is the ATP synthase, an integral protein consisting of several different subunits. This proteins is directly responsible for the production of ATP via chemiosmotic phosphorylation. It uses the proton gradient created by several of the other carriers in the ATC to drive the mechanical rotor. The energy from that rotor is then used to phosphorylate ATP to ADP.